Hi, everybody. Welcome to our Lunar Lab Tour. We want to welcome you, all of our interns that we have. We have interns from all over. We have interns from LPI, Super, the Exploration Summer interns. We have Jacobs and ARIES interns, as well as our NASA and our co-op interns joining us today. So we have some great speakers for you that are going to be talking about our Lunar Lab. We have Kim Willis, Suzanne Foxworth, myself, and Juliane Gross, and then we also have Paige Graff, who's going to be with our camera and helping do questions. So I am going to turn it over. Please make sure your microphones and your cameras are both off. And if you have questions during the session, please go ahead and put those within the chat. And then we'll do a Q&A after the program is over. Thanks.
Hi, I'm going to do a quick introduction of the people who are part of this or bringing this tour to you. My name is Kim Willis. I have a bachelor's and master's degree in physical science, concentration being geology from University of Houston, Clear Lake. I also have a master's degree in secondary education. I began my career here at Johnson Space Center, right in that very lab that you're going to be touring today. And I worked there for many years, and then I decided I needed maybe a different view of the Earth and processes. So I started working with the Space Shuttle Earth Observation Project, and I also worked with the Crew Earth Observation Experiment on board the International Space Station. Basically, I briefed astronauts and directed them what places on the Earth to take photographs. Currently, I'm the contractor manager for Astromaterials Curation, basic and applied research and outreach and data management. And this is one of the really fun things that I get to do with my job. The other person that you heard from earlier is Suzanne Foxworth. Suzanne obtained a biology degree, a secondary education degree, and a master's in physics education from Texas Wesleyan University. She was a classroom teacher for 15 years, give her a big hand, and has been working as an ARIES communication and outreach specialist for the last 7 years. Suzanne is also one of the coordinators for NASA's Lunar and Meteorite Sample Disk program. You'll also be hearing today from Dr. Juliane Gross. She completed her master's degree in petrology in 2005 and a doctorate degree in 2009 in experimental petrology from the Ruhr University, Bochum, Germany. And following, she went to graduate school where she earned a postdoctoral fellowship at the Lunar Planetary Institute, where she transitioned her research from terrestrial sample science to planetary sample science to work on lunar meteorites and Apollo samples, and later worked as a research scientist at the American Museum of Natural History in New York City. In 2015, Juliane moved to Rutgers University, where she's an associate professor for planetary sciences. Since 2019, Dr. Gross has been the deputy curator for the Apollo samples at NASA's Johnson Space Center in Houston, Texas. She has led the preliminary examination of the ANXA Apollo Next Generation Sample Analysis program and is also involved in a multitude of Artemis mission-related projects. Along with that group is Paige Graff. She's the lead for our science engagement. And you may be hearing from her voice, but she will definitely be assisting in the lab with Juliane with the camera. So we talk about ARIES. What exactly is ARIES? ARIES is Astromaterials Research and Exploration Science. So ARIES does a lot of things, but basically they want to advance human space exploration, to integrate terrestrial and planetary research, and to promote successful space missions by mitigating risk. So there are many groups that do many things to help meet these mission statements. Today we're going to focus on ARIES Curation, and within ARIES Curation we're going to focus on the Apollo collection. Now you're going to hear us say Lunar and Apollo interchangeably, but we're trying to change mostly to Apollo because, as you know, we have Artemis, the next mission to the moon. And when we get samples back from the moon from the Artemis mission, we want to be able to tell the two apart, because if you just say lunar, they'll both be lunar. So in ARIES Curation, we protect, preserve, and distribute samples to scientists, including the lunar, the Apollo rocks and soils, Antarctic meteorites, microparticles from the Stardust spacecraft that collected from a comet, and high altitude collections, and atoms from the sun, which was the Genesis spacecraft, and including space exposed hardware. If you would like to explore further or in more in depth of what ARIES does, we have the URL here at the bottom, aries.jsc.nasa.gov. So we had 6 missions that went to the moon. And with Apollo 15, they had a lunar rover, which enabled the astronauts to go farther from the lunar module that you can see on the right, so they can collect more samples from different areas. So on the left, you have Gene Cernan and in the lunar rover, and on the right, you have John Young, who is the Apollo 16 commander, and Gene Cernan was the Apollo 17 commander. Now, this is the building from the outside of which you're going to visit today. So there's a few things that I wanted to point out. One of them being that this floor, you'll see the steps that are coming up. And those steps are a half a floor higher than the buildings at JSC. And the reason for that is they wanted to build this building, which was completed in 1979, to house the Apollo samples, but also to be protected from hurricanes, which we have here. And in fact, the building was designed to withstand a Category 5 hurricane. And also, you know, hurricanes often have storm surge. So that's why they wanted it to be a half a floor up. Now, at the very top of the building is where the Lunar Lab and the Lunar Vault is. Now, this is a very special display cabinet where we show rocks from different missions, and there's a few things going on. It's kind of a busy slide, and so I'll tell you about a few things. One thing, you're looking into the lab from this display cabinet, and each cabinet in the background that you see have Apollo samples from different missions. But the display cabinet that you see facing you has windows on both sides, and you'll notice that it has rocks from different missions. It's the only cabinet where they will have rocks from different missions that are open. This lab is an ISO 6 clean room, and what that means is you can't have more than 1,000 particles that are greater than or equal to 0.5 micrometers per cubic meter of inside air. Also, inside these cabinets, you can't see it, but they have nitrogen gas. Why nitrogen? Well, because nitrogen doesn't interact with the samples, which is a good thing. We also have positive pressure in the labs. That's why you see the gloves and you see these covers over the gloves. And that's because of positive pressure, those gloves would be poking out if we didn't have covers on them. And the reason for that is if you had a hole in the glove or something happened, then the pressure on the inside of the cabinet is greater than the pressure on the outside. So the nitrogen is flowing out. The oxygen and the moisture is not flowing in. There are only specific things that are allowed to come in contact with the samples. That would be stainless steel, Teflon, and aluminum. The other thing that you can see here are photo cubes, the little square cubes. You can see a T or a B on some of them. That's because it's top, bottom, north, south, east, west. Now, a few of these samples were in the actual orientation that they were on the moon. Most of them were actually an orientation that was given to them when they were returned to Earth and opened in the lab. And the reason for that is that when you break off a piece or you bandsaw a piece, you want to be able to orient that piece back to the original sample, and then you can tell where exactly this sample came from and what side it was on. Also, if you'll observe, there are some flat surfaces on these rocks. That's not natural. Those occur because we take them into a different part of this lab and we bandsaw them. So we saw them we saw pieces off of them, and that's why it's flat. The other reason that you have these rocks in there is because they show the basic kinds of rocks that were brought back from the moon. So on the left-hand side is an impact breccia. So it's a rock that was created from an impact. So the little white things are pieces of broken rock that, because of the temperature and pressure, was then compacted to make a new rock. You have basalts. You have one there in the middle. It says 15016,0, and the one on the end as well. Those are basalts, which we have basalts here on Earth. They look similar, but because they're from the moon, of course, they're very different. And then the one that's white, that's a highlands rock. So when you look at the moon at night, you see the bright areas. Those are the highlands, and they're made of this white rock, and it's mostly made up of anorthosite. And some of those lines that you see are caused by the saw going through the rock. It heats up and it actually changes the color. The dark areas of the moon that you see are areas that were filled with basalts, and they're dark. And so you can see why that's 15016, and the 1 on the end. That's why you have the dark color. We also have monitors inside these cabinets. We are monitoring them 24/7 for any moisture or oxygen, because obviously we want to keep these samples as pristine as possible, and we don't want anything to get in. Now, this is the lab, and you see the contractor staff that are working in there. And I want to make the point that actual research, for the most part, doesn't happen in these labs. Mostly what people are doing is preparing these samples for other scientists. Now, the question is, how do samples get in and out of those cabinets? If you look at the cabinet on the right, you see this sort of cylindrical tube. All the way around that lab, you can see the windows is a corridor. So you open up the outside door, the inside door is closed. You cut off the outer bag, you put a glove on your hand, you put in the sample, or if you're putting in tools, we never put in samples and tools at the same time. Then you put it in that tube, you close the door, and you purge it with nitrogen for 5 minutes. Then you open the inside door, and then you can bring things in. So the reverse is true when you want to take things out as well. And this is a closer view of the staff that works in there. On the left is Andrea Mosey. She's the contractor lab lead. And she actually started out as an intern here at Johnson Space Center. But she's been here for a while, and anything you want to know about the Apollo collection, you can certainly ask her. Standing to Andrea's right is Charisse Kreischer. Charisse was the person who got to open a core, an Apollo 17 core, the first time that's been done in over 20 years. So Charisse was the one doing that very meticulous work. In the middle is Lacey Costello. She's the newest member of the Lunar staff. To her right is Linda Watts. Linda has also been here for a while, so if you want to know anything about procedures or the history of why we do things, Andrea can answer, but so can Linda. And on the right is Jeremy Kent. And Jeremy not only works here in the lab, but he also makes thin sections out of the lunar samples. Not in the lab that you're looking at now, but that's in a different lab. Basically, everyone has a degree, either a bachelor's or a master's in geology, but Andrea, her bachelor's was in math and chemistry. So that doesn't mean that you have to have a degree in geology to work in this lab.
So we are very lucky today to have Dr. Juliane Gross in the lab with Paige, and she's going to take us on a little tour inside the lab. Take it away.
Whether you can you all hear me and see me? Hello. All right, I'm getting the thumbs up. Great. All right, well, welcome to the Lunar Lab. This is the Pristine Sample Preparation Lab area that you can see here behind me. There are all these different cabinets here, and I'm going to take you on a little tour, and we're going to start with this lab, and then I'm going to take you into the ANGSTA core lab, and then we're going to go into the Pristine vault in a little bit. All right, so here's an example of one of the typical Apollo cores. Apollo cabinets. You can see here, there are little marks. So this AP17 stands for Apollo 17. So only Apollo 17 samples get processed in here. So we never ever mix different missions because we don't want them to cross-contaminate. And Kim just talked to you guys a little bit about it. And so when we look inside the cabinet here, you can see that we're in the process here of processing some of the samples for a lunar PI, the principal investigator. You can see the original grained rocks here in teflon with their number on it, and then we split them into smaller pieces, and you can see that in the tray, in the aluminum tray over here with the little aluminum cups, and you can see that we started labeling each cup. And so that's a comma 184, so each time we split a rock into smaller pieces, each piece gets another number. And so we have here 78235, and then that's comma 0, so that's the mother rock basically. And if we cut that, then it becomes comma 1, comma 2, And so, 184, that specific rock has been split 184 times, and so on. All these cabinets positive pressure with nitrogen, and so you can see here, if I increase our cabinet pressure here, so you can see the nitrogen flow right here, right? And then if we open one of the gloves, they're just gonna stick out, and so that we know we have positive pressure, and if we want to work in these, we just have to like stick our hands in here. I'm not going to do that because it's a pain. I'll show you when we go into the core lab. But we do this so that the Apollo samples never ever get in contact with Earth's atmosphere. So this lab here is basically, it was built as a clean 1,000 lab, which means no more than 1,000 particles per cubic foot. But we are actually really careful. We have to go through like an air shower, which is not a shower like you see in the meteorite lab, where you have like jets that blow on you. This is a laminar flow, because if you have these jets blowing, they will just stir up particles, and they're not really removing the particles. And that's good enough for meteorites, because the meteorites have already seen Earth's atmosphere and sitting on Earth's ground. So the meteorite lab is not as clean as the lunar lab. And so when we go into our air shower, we have to stand in here for a minute, and we get a laminar flow that very carefully flows over you and like gets sucked at the bottom. And so we do particle counts every week, and that shows that this is actually a clean 100 room, which means there are no more than 100 particles per cubic foot. And so that's great. We have nitrogen in these cabinets to keep the water out, because we don't want the rocks to start rusting and get contaminated with like Earth atmosphere crap. And that includes organics. And so we have one of our curators who comes in and does biological swabbing. So they come and swab all the surfaces, and then they try to grow bacteria or viruses or whatever that might be in the lab, and then that will tell us how badly contaminated or how great our lab is. And depending on what the results are every month, we then maybe change the procedures. All right, so this is over here. This is the only cabinet where we mix samples, and this is the cabinet that Kim showed you. And if I turn on the light, you can see it's empty, because guess what? It's hurricane season. And we had to bag samples in teflon and move them into the vault, because we don't want them to sit out here and then something bad happens and we might lose power, in which case our nitrogen flow goes out, and then there's the possibility of contamination. And so when hurricane season is and there's a threat or something, we will bag all these samples and put them in the vault so that they're all safe. And so that's why the cabinet is empty at the moment. But hopefully maybe next year, when we can all be in person again, and maybe you all come to LPC next year, we can organize maybe a little tour. We can go over there in the VVA, which is the viewing area, where you can see, and then maybe we have the samples out and you can actually see them in person. I'm not going to promise anything, but it is a possibility. All right, so now I'm going to take you into the core room, and that's where I mostly work at the moment. And that is a very special room with a special cabinet that was built specifically for the ANGSA program. And so that is the Apollo Next Generation Analysis Sampler Program. All right, come on in here. And so almost 50 years ago, when NASA brought back samples, and this is from an Apollo 17 sample, they had the good foresight to keep some of the samples sealed. And so in 2019, NASA decided it's time to open one of these sealed samples. And then Charisse Kreischer and Ramosi and myself, we were the three women who actually got to open this sample that, you know, nobody has ever seen, nobody has ever opened it before. And so this is a special cabinet that was built specifically for that. And I'm going to show you the sample, which is right here. I'm going to come over here. So this is the core sample. And so we have dissected the core. And so here, hold on. Okay, this area is the top of the core, and this area is the bottom of the core. And so this was a hollow aluminum tube that the astronauts hammered into the ground, and then the tube filled with rocks and soil. It's a double drive tube, so it's twice as long, and then they sealed the bottom part and they sealed the top part, and we just opened the top part. Now, when we do this, we are hoping to see different layers underneath the surface that we otherwise would not be able to see. And so when we opened the core, you can see the receiver sticking out here where it got pressed. We started dissecting the core, and so we took layer after layer off, and this is the last layer that you can see here. And I don't know if you can see this, but the top of the core, let me see if I can move my hand so you guys can see the top. This is the top area. If you look very closely, it is darker than the bottom of the core. Here, where's my finger? Here, you can see this area here. See how this is so much darker than the rest of the core, which is below the surface? So this was on the surface of the moon, and it's so dark because this is space weathering. So when you have material or grains or soil sitting on top of the lunar surface, there's no atmosphere. So you have solar radiation and bombardment happening, and that changes the properties of the soil. It makes it darker because it puts nanophase iron in there or converts it. And so we can actually see the actual space weathering happening. And so a lot of researchers are interested in these space weathering products because we can then translate that to other airless bodies. And that's what makes this so important. And because this core has never been opened before and it's like super pristine, when we opened the core, the first sample we took was some of the space weathered area from the top and some of the pristine area at the bottom. And then that got sent out to researchers who are looking for organic materials from the moon and also different D to H ratio from the moon to maybe learn something about space weathering. You can also see some holes in the core towards the bottom. See that? If I move my finger, there's a hole. And so there was a big chunk of rock that we pulled out, and so it left a little cavity in there. And so we have dissected this core a lot. And so this rest that is left over is going to get epoxied, so impregnated, and then we're going to cut it into thin sections so that we can like look under a microscope and do all sorts of petrology and geochemistry on it. And then after the science PIs have done their studies that they got funded for, we're creating this preliminary examination catalog, and then everybody on this entire world can request samples and work on this. And so over here, I'm going to stick my hands in. And like working in these cabinets is really difficult because you have these massive gloves on. And as you can see, it also is restricting movement. So if I want to grab something from the shelf, I have to start do yoga so I can like reach up higher with my arm, right? And so we have different things here. This is a heat sealer. So when we take samples, they are put in little containers. I'm going to show you some of the containers right here. So here, these are the little containers, the lid. So the sample goes in here, we put the lid on, and then we bag them in Teflon bags. And so each sample gets 3 Teflon bags that we seal in here. And only once they're sealed 3 times, we then put them in the airlock and remove them out. And we do this 3 times just in case the outer bag is leaking or maybe the middle bag is leaking. Then we have 2 more contingencies just in case. So we really try to keep all the samples safe. You can also see some of the foil that's up here, there. And then you might see, if you can see this over here, there's the foil with a little black wafer. See that? Right here, this thing. So these are witness plates. And so we have those in there, and they collect particles. So when we give lunar PI samples for organic measurements, we want to make sure that their results are really from the moon and not by accident contamination from Earth. And so these witness plates and witness foil, whenever somebody gets an organic sample to look for organics, then we also send a little piece of this witness foil. And so if they happen to find the same organics on the witness foil, well, then their results might, indicate contamination rather than organics from the moon. All right, so let me put the gloves back on. All right, and we just do this so it's nice and neat. And then I'm going to take you into the sample vault. So all the samples that we have taken from this core, we have allocated some to the PIs, and the rest went into the Pristine sample vault. And so that's where we're going right now. Okay. So the fun part is when you work here, even if you wake up in the morning and you have a really bad day and you come into the lab, you're like, okay, everything is great again because you actually get to work on the moon, and that's just the best thing ever. All right, so now we're going to go back in the air shower. But because we're going from a clean environment to another clean environment, we don't have to stand in there. But if you point this up, you can see the little holes where all the air is flowing out. So technically, we're getting flowed right now. All right. So now we're in the corridor behind the Pristine lab. And so you can see these things here. These are airlocks. And so that's another one here. So when we take samples out of the Pristine sample vault and they're still in their bags, we put them directly into these airlocks, and then they get flushed with nitrogen. And then the other person on the other side pulls them into the cabinet. So the samples never, ever get to actually be outside in the sample lab, even though it's a clean lab. We still don't want them to be in the big lab out there. So they get directly transferred into these airlocks. And so you can see here how the airlock connects to the corridor. There's the cabinet. And so one person would be on the other side, and then after purging about 5 minutes to really push all the earth air out, we can then pull the samples into the cabinet. All right, and I already opened the vault for you guys. So this is our vault. You can see that if you come over here, we have 2 different sets of combinations. Nobody has both combinations for security reasons, and so we already opened it. And this is the same vault door that the Federal Reserve Bank had 50 years ago. So when this facility was built, they went to the Federal Reserve Bank, had the state-of-the-art security vault doors, and that was the same door that we got. And so now we're going to go in, and this, welcome to the Apollo Pristine Sample Vault. This is where every pristine lunar rock is being stored that is not currently out with investigators. And so you can see all the cabinets, again, are labeled. For example, here's an Apollo 16 cabinet. And you can see all these trays in there, all the trays. You can see the little metal tags on there, the aluminum tags or stainless steel tags. Those are sealed. So every time we have to go in and pull samples out for a researcher, either Ryan Ziegler, the curator, or me as the deputy curator have to go and read down the number, and then we reseal it, and then the trace out, and we get the sample that we need to transfer the sample to, and then our processors then can take the samples in the cabinet and break them apart into smaller pieces, and then they get sealed and sent out to the lunar PIs to do their research on. Now, the cool part about this vault is there's an area, a high secure area, and you can see these fabulous high-tech security system right here. There's also a sign which unfortunately fell off, but you can see how high-tech security this is. So this cabinet is special because I told you about ANGSA and the Apollo Next Generation Sample Program and how we opened the first part of this double drive tube, right? Now the second part is still sealed, right? And our plan is to open that hopefully this fall. And so you can see that when it came back from the moon, it was vacuum sealed again. And so technically, we're hoping that the sample was sealed under vacuum on them and then put into another vacuum container on Earth. And so what we want to do is sample the head space and see what kind of gases came out of the core before we open it and then dissect it just like we did with the other core. And so that's super exciting, and unfortunately the pandemic has put us way behind schedule. But fingers crossed it's going to happen in the fall. We're moving forward with the gas extraction manifold, and ESA is helping us build stuff, and so this is the multinational project. Yeah, so I don't know what the time is, but this is okay. This is the big lab here. Basically, there's still some space. So when Artemis happens, there's enough space that we can put more cabinets in here and store the Artemis rocks here if they go into this lab, which we don't know yet. Or maybe we'll build a new lab where they go in. But there's enough space here where we can do things, and we can have a whole new area for the Artemis rocks from the moon. And there's also space in the Apollo sample, so we can consolidate some of them. All right. What? Oh, I actually don't know, but it's super heavy. There's a lot of pressure. There. And then we spin the locks. And then we'll sign the door so that security-wise we know that we locked it. What time is it? 1:35. And then we sign this. And that's how we keep track of how often the vaults were opened and by whom it was opened, just in case, something goes missing ever. So if you guys were here in person, I would let you spin the locks but next time. And then here in these cabinets that you can see, that's where we store a lot of the tools that we need. Here are some of the teflon cups where we put samples in to look at them. Here are some of the containers. And so all of this is backed in Teflon. You can see scissors and stuff. And so everything gets cleaned and then pre-cleaned and then cleaned so that there's zero contamination. They're all sterile. And then they get backed in Teflon. And so then what happens, you can see we remove the outer back because it has been in contact with air. And then the inner back still has the scissor. And then we put the scissor with the inner back into the airlock. It gets purged. And then when the scissor gets put into the cabinet, then the processor will open the last bag and pull out the sterile scissors to be used. And yeah, so this is where we get a lot of our tools and equipment from. And that's it. So when people work on the Apollo samples and they open them in their lab to normal air and do research on, whatever is left over, they're going to bring back to us. And then we cannot reintroduce them into our Pristine lab. And so we have a return side where we process return samples. And so that's what you can see in here. So that's the return side. There's a vault over there as well where we keep the return samples, similar to the Pristine vault, but not quite as clean because those samples already have seen Earth's atmosphere. And so this is the returned lab, processing lab, basically. And you can see that instrument that sits on that table, that's the gas extraction manifold that was built so we can start extracting the gas of the next core. So Charisse, Andrea, and I have been already practicing and going through procedures, and we're excited. So hopefully, fingers crossed, this is going to happen in the fall. You never know, so always be prepared for the unexpected. But hopefully we don't have to at this point. All right, so we're going to go back in the lab. Yeah, so over here, these are the little cupboards. You can see all the blue one, that's my sweater right there. So when we go in, we have to change. You cannot wear jewelry or anything. So we leave the stuff in the cubbies, and then we take the clothes. Everybody has an assigned little cubby area, and then we get fully dressed. Once you're dressed, we stand in the air shower just like we did. Oh, and look, there's Jeremy. Hi, Jeremy. So he's going to come and process some samples. And now we're going to make him uncomfortable staring at him. Yay. All right. Why don't we let him change? That's too funny. So, yeah, actually, Jeremy is going to work on the Apollo 17 samples here because he started this work. And so when one processor starts on a project for a specific lunar PI, we never change out the processor. So if you start a project, you will also have to finish it because every processor works slightly different, and we don't want to introduce another source of error by switching out people. And so that's why every processor gets assigned a specific project, and the curators, like Ryan and me, we write these curatorial orders so the processors know what to do, and then they go ahead and do it. And they do a fantastic job. We couldn't do this without them, so we're super happy they're here. So this is a really cool cabinet. This is Apollo 16. And so here you can see that we digitize things, and so there's a monitor here. So if we have samples on here, we have a microscope, and then we can display whatever we see here on the display here, the sample, which we don't have any samples in right now. So here's a different light source. You can see this flickering on. So if we had a sample here on the photo jack, you could move it up and down, and then you can move over the processing samples of taking pictures. And so this cabinet we started to digitize, so it's going to make the life of the processors easier, and it's more precise. But again, it's hurricane season, so we don't really have anything out at the moment because we have to be a little careful. And there's also a pandemic happening, so yeah. All right, well, I think I'm going to give it back to Suzanne and Kim and I'm happy to answer some questions if you guys have any. I'm just going to turn off the lights real quick.
So we do have a few questions. One of them is, what kind of things do you prepare to open the core?
So before we open the core, we had a mock-up glass box. Jeremy's making funny faces in the air shower. We had a mock-up glass box, so we could practice, and we had a mock-up core with simulant, filled with simulant, so it wasn't real. And so we had to practice how to open this, how to put the equipment together inside the cabinet. And so there were months and months and months of preparation. And we also had to learn, the three of us, how to communicate with each other while we are on the cabinet working in different areas. And so Charisse, who is the lead on this, if she needs a screw or needs someone to hold something, she can't say, hey, hold this, because when I have my hands in the cabinet, I can't see what she's doing. And so we had to learn how to communicate correctly with each other to make sure we're not by accident, destroying the core or do something that the core falls apart or something. And so there's a lot of practice going on in the beginning of how to build the stuff, how to extract and open the core, how to then extract it onto the table, and then also how to do the dissection. There are lots of procedures that have been written 50 years ago, and so we had to go through all the procedures and update them. We had to test the existing equipment to make sure that everything was still there, and if it wasn't, then we had to find the old manufacturers and to make sure that we can still get those parts that we need. And so there were months and months, 1.5 years basically before we actually opened the core of preparation work that was going on. So practice is a good thing. Nobody, falls out of the sky and is a master in dissecting the core. It's a lot of practice that goes into this.
Another question is, how do you store or keep track of dust samples?
So when we open the samples, they are a little dusty. So there's different grain sizes, and we can sieve the soil into different grain sizes and then store them in containers for different grain sizes. And so when you open them, you have to be very careful because they're very static. So if you open them and you have the teflon, sometimes these grains jump. But that's why we contain everything in one mission. So each cabinet is just one mission. So if there is, let's say, some of the dust grains fall out, then we can sweep later on the cabinet, and everything that got mixed gets put into a different sample container that has like a 9099 number. So we know these are, this will call cabinet sweeping. And so that means the samples from the same mission got mixed together a little bit. And so we store them slightly separate. But we'll try not to lose too many grains or dust, but sometimes it's unavoidable because they're just statically and they just jump.
And another question is, why do you use Teflon?
Oh, so we don't want to contaminate the sample. So if we use any other material, not just like plastic, that has organics in it. And so then you're contaminating your sample with organics. And if there's a person who wants to look for organic material on the moon, you're already handing them a sample that is potentially contaminated because we put, organic material around it, like normal plastic, which has contaminants and stuff in it, organic contaminants in it. So Teflon doesn't. And then we can use the Teflon also as witness. And so we can send them a piece of the Teflon, or the sample gets sent in Teflon, so they can test the Teflon and test the sample. And so if they get different answers, and they know what they found is originated from the moon and not necessarily from Earth.
And do you have any thoughts or advice for the interns?
In the sense of career, yeah, I guess so. If you, I think my big advice that I would give students is, whatever you pick, the topic that you pick or the field that you're choosing, choose something that you like. And if you later on learn that you don't like it, you can switch fields, right? I switched fields. I did my master's in metamorphic petrology, where I looked at continent-continent collision zones. And then in my PhD, I did hydrothermal diamond anvil cells, looking at subductions on fluids. And then after I got my PhD, in my postdoc, I completely switched fields and went planetary science, right? And so just remember, you're never stuck in a specific field. There are always ways to switch. The fun part about being a scientist is that you continue to learn, right? That's what we're all passionate about. We like learning, and we like learning new things, and we like exploring. So my advice would be, stay curious, have fun. Don't ever give up, right? Grit is a big part of being a scientist. Stick with it, and then just enjoy what you're doing and enjoy life, because not everybody gets to work on these precious samples, and any astro material, no matter where they come from, which body, are great. You're basically being an astronaut without actually leaving Earth, and that's the part that I really like best about this. So yeah, just enjoy it. It's awesome.
So we had a few more questions come in. One, they want to know how the lab is preparing for the 16th Psyche metal asteroid mission next year.
Oh, so we're building another part of this building. So those samples don't come. In this area, we only store Apollo samples. And so there are different labs at JSC that store different samples from different regions in the solar system. So Genesis has their own lab. OSIRIS-REx is getting their own lab. And so all the sample return missions are getting their own lab spaces because, again, we don't want to cross-contaminate samples, right? And so here, we're only storing samples from the moon, and only the samples that were returned by astronauts, right? We have meteorites that come from the moon, but they go into the meteorite lab. And you have different meteorites from different solar system bodies, but they can all be stored together because they all have already been sitting on Earth, happily for however long. And so each mission gets their own curation facility to keep them safe, so we can then allocate samples. And that's how we prepare for these.
Okay, and this one's going to be kind of long. I noticed the ruler beside the core sample. How precisely do we know what depth each part of the core came from? For example, does the regolith get compressed at while the core is being taken, or by Earth's gravity, or is the shift while the core is being opened?
That is a really good question. And so the way this normally goes is you have these hollow tubes, and the astronauts hammer it into the ground. And we know how long the tubes are. So if they fully fill, right, they do compact it a little bit, but not that much. And then you put a cap on, and so then we know that the core full length, let's say, was like 70 centimeters or something. And so we know it's 70 centimeters below the ground. The moon is actually quite compact, the surface. It's not that fluffy or dusty or something. The problem with this particular core was when the astronauts took the upper part, when they took off the upper part, they held it up like this, and then part of the core actually fell out. And so if you look at the Apollo 17 images in the movies, you can see it fall out. And so when the core came back, what they did in 1972 is they took an X-ray of the core, and you can see there's a corner where there's material missing. So when we prepared to open the core, we wanted to CT scan the core. We wanted to do X-ray computed tomography, kind of like when you go to a doctor and they scan your brain and see if there are any tumors, and they make a 3D picture of your brain without actually cutting you open. We can do the same thing with rocks. And so we scanned the core tube to see what's in it. But in order to do that, because then the tube gets scanned like this, we had to compress it a little bit so that it's safe and things don't move around. And so originally the core was, I think, 23 centimeters long, and we compressed it to 18.5. But because we scanned it before, and when we extruded it, we actually compressed it a little bit more. But now that we have the original X-ray image and the CT data, and we know when we dissected the core, we do every half centimeter is one interval. So we very carefully dissect each interval. We can now go back, and each little rock fragment or class that we find, we can then refer back to the original image. And so we can calculate where the compression happened, whether it happened on the surface, or where it happened somewhere in the middle or somewhere at the bottom, or whether it happens around big clasts or small clasts. And so that is all part of the preliminary examination that we're doing to get the sample catalog out, so people can understand better where the different intervals are and what they relate to on the surface of the moon, whether it's 1 centimeter below the surface or 70 centimeters below the surface. And so there's a lot of calculations and correlations going on.
So we have this one. What is your favorite discovery or finding?
Oh, that's a hard question because I like a lot. So my favorite moon rock are breccias. And they're basically rocks that are composed of different rock types. And all these different rock types come from different regions, and they all get baked together into one rock, basically. And so you can think of breccias as the treasure troves of the moon, because you can learn a lot about these different rock fragments in there in one rock. My favorite discovery, I think, are rocks that contain spinel. And so that's an oxide, magnesium aluminum oxide, and the color is pink. And so we have some rock types or some rock fragments in lunar samples that have these little pink minerals in it. And number one, it looks pretty, but number two, it also gives you an insight of like how things have reacted on the moon. So what we think happened is that you had a big hot magma chamber, and magma coming up and then reacting with the crust, with the amorphous of the crust, and that reaction produced these pink spinels, and they settled out, and that's how you make a new rock type. And then a big meteorite impact happened and excavated the material, and then part are still in the crater, so we can see this rock. It's a new discovery with remote sensing missions, but we also have little pieces in these breccias. And we also have found some of these little pieces in Apollo samples. And until a few years ago, we didn't know that this rock type existed to that extent. And so that, to me, was super exciting because I was part of that discovery. So I think that's my favorite part.
And then also, if they do build a new lab for the new moon missions, is there anything you would think they should do differently?
Oh, that's a really good question. I think that with building the Apollo lab and keeping the samples pristine, they have done a really, really, really good job. So I think if we build a new lab for Artemis, then there's a lot of things that we should do exactly the same, like keeping the cabinets under positive pressure with the nitrogen flow in there, keeping missions separate from each other. All of this needs to be repeated. And so these samples were stored under like the best conditions possible. And so if they build a new lab, I would hope that they would do the exact same thing, and hopefully also be as restrictive with the materials that we can have in the lab as we have done with Apollo. Hopefully when Apollo came back, we didn't know whether we need to protect it from the samples or the samples need to be protected from us. And so in the beginning, we were protecting ourselves from the samples because, you know, we wasn't sure whether they have like some kind of like, I don't know, weird stuff in it that could kill us. And so the very first samples that came back, they were maybe not kept as pristine as possible. But now we know there's nothing on the moon that can harm us. And so once our Artemis samples come back, then we know we can see them on the moon, store them sealed, and then once they're back on Earth and transported to JSC, we can keep them even more so pristine than we could the Apollo samples. And so technology has evolved, and so hopefully we can really seal them and keep them in good containers until they're in the pristine lab, and then we can store them in the vaults. But Apollo has done a really, really good job with keeping the samples safe and pristine, and so I'm hoping that we do the same thing with Artemis.
So I want to thank all of the interns for joining us today, and thank you, Dr. Juliane Gross, for the tour, and thank you, Paige and Kim, for all of the
just hold on,
insight.
I need to say one more thing. So I know you all have seen the meteorite lab, right? Just want to remind everybody that Lunar is better. That's all I got to say.